zero here?
Acanthonus.
Yeah.
Acanthonus. Acanthonus. Google. Spell that.
Yeah, okay. Yeah. Yeah, I'll swing over a little bit. Just because we're going up so much, I might just try and tilt up a little bit.
I don't know if we've seen, if we want to talk about something rare, that one coral that like only sticks on the wall, and we tried to sample it with the slurp gun.
Oh yeah, that white coral looking
I feel
thing.
like that's the first time, and
The,
last time
the
we've
one that
seen
was like growing in strands.
Yeah.
Yeah. Yeah.
The ribbon coral, or
Yeah. Yeah. That was an interesting collection. I'm happy we got that one. Class sponge, Euplectillid.
Go ahead and do a partial zoom here.
And then we saw the Dumbo octopus as well.
Oh, yeah,
Possibly
Dumbo octopus.
Dictyalus.
Dictyalus.
Got any more zoom?
Sure do.
Now that I say that, I get off my head in here.
This was collected previously in 2017.
Sponge.
This area.
Yeah, does it just look blurry, or
Yeah,
are we out of
yeah,
focus?
no, we're in focus.
Wow.
It's
Oh, it's got a weird optical illusion, yeah.
All right, you can go ahead and combine it.
Sure.
Super cool.
It's quite long.
Yeah.
Answer the question in chat. If you find new species, what would you name it? Technically, we're not allowed to name it. It goes through a special committee of scientists, and then after years and years and years, apparently, they all get together and decide on a specific name for it in Latin that's super long and very hard to spell.
sometimes in Hawaiian too, though.
Sometimes in Hawaiian, and in other languages if it's native
Yeah.
to that country's area.
Yeah, I think they get together another committee of indigenous scientists that would collaborate with other scientists researching the organism. Steve, you have some experience in that, right?
So it's not quite exactly like that. You know, there's a lot of work that has to go into species naming and descriptions. It takes years because you have to make sure that that collection or that species that you've identified is not anything that's been described anywhere else in that taxon. So for example, if the remaining specimens from that taxonomic group, genus or family exist in museum collections, you usually have to go through and make sure that that is distinct from what you have. So that can take some time, either requesting specimens or consulting some of the literature that's been published on that subject. After that, typically you have to prepare some proof, some evidence that it is unique. So for example, we use a bunch of different types of tools, from different types of microscopy to sometimes genetics that will further show that that species or that taxon is distinct from anything else that's been recently described. And really the naming comes at the very, very end, and it's usually, almost always, follows the rules of the International Code of Zoological Nomenclature, which is the authority. But it's not named by a committee of scientists. It's usually just the scientists who author the publication. And typically, even in a publication of several authors, really only a few might be the authority on the taxon. So they are the people that can identify it if anything similar is brought back. So yeah, there's several steps, but yeah, the naming convention is it's sticky. Yeah, it's the rules don't like to be changed very much, but that helps us keep the kind of the rules fairly consistent. But yeah, they have been stretched in recent years.
And what about renaming of organisms? Does it go through a similar process?
So you can re-describe something if it doesn't fit taxonomically in the group that it was assigned to anymore, or if further evidence has come to light that suggests that it's not any longer related to that. A lot of that happens sometimes when older specimens are sequenced or new genetic materials are sequenced and we start to apply. Can we zoom on that yellow blob?
Sure.
Top middle. I believe it's an urchin.
Blob.
All right, go ahead and start your zoom there, please.
You got it.
Wow, Steve, you have
Nice.
good eyes.
Steve has good eyes.
Go ahead and fish on in a bit more if you like.
Sure.
Ooh, a little shrimpy fishy thingy.
Come a little way.
Yep.
Thank you. That's where I put it. Go ahead and fish on in a bit
Middle
more.
of the Pacific.
Pull away, please. I mean, someone really had hit that pretty hard for that's a tennis ball, you know what I mean?
Looks like a tennis ball.
Yep. Yeah, so, but only if redescriptions are warranted, can they be, redescribed, in a publication. It still takes a while to put together a publication. You just can't go about renaming species because taxonomy is a process that relies on, a structure and, a priority and a hierarchy. Otherwise, it's very easy to lose, the origins of names sometimes if you can't trace it back to what we call the type specimen or the original thing that identified that family or that genus
Like
or
specimen
species.
0.
Yeah.
Gotcha.
Yeah. And that's why type specimens are actually guarded like some of the most valuable items in the world, like usually in museum vaults, for example, because they are the authority. That is the thing that's how we know that all things descendant from that name are linked to it. And so, and when those type specimens are lost, that creates a big problem. A couple of years ago, there was a massive fire at a natural history museum in Brazil. Does anyone remember this?
Yeah, I
I heard
remember
about
that.
it. I didn't know what
I'm just
happened.
going to
though.
swing over to
Many
090.
type specimens were
You wanna
lost
hold it.
from
I like using
Amazon
your sonar on the
biodiversity.
head.
So
There we go.
that was a huge blow.
You'll be out of
So then what
image at
happens then?
070
Do they
right
just
now.
do their best?
It's okay.
Well, you hope that there are what are called paratypes available. So paratypes might be the same type of species stored elsewhere, but doesn't have the type priority, so it's not the first. But it might have been described at the same time as the type specimen.
Just going to bring it to 075 to catch
But
you.
it's very important to kind of maintain that link, because sometimes some of these specimens were described as far back as the late 1700s, and to maintain that continuity is really valuable.
Cool.
A little valley ahead of us here.
Then to answer that question in chat, have you found any gold? I mean, for science,
No
gold.
elemental gold.
No elemental gold, but video gold.
Lots of video gold.
I'd be extremely surprised if we
Keeping
were
our
to find
delta
gold
low
in
just because you're
this
so
geologic
far ahead.
environment.
Right?
These the crusts don't have gold as their composition, or is it?
It probably
Low?
has like really low.
Yeah.
So if you want to call that gold, but I
There's a
assume they
little
mean native
critter
gold.
below your bubble can there.
Which no.
Little fish.
Usually you find that elsewhere. I'm not an ore guy, but
Current heading is 071.
Are there possible to
Roger.
find like rare metals and stuff like that at this depth? Or would you have to go a little deeper to find them?
it doesn't matter how deep you are.
Really?
Yeah, the ferromanganese crust has rare metals on it.
The topography
It
is
is
cool.
rare metals.
All right.
Nickel,
Do a
cobalt,
partial zoom in.
rare
right
earth elements.
Sure. Anything in particular you want to look at?
here.
Those
There's
concentrations are all found.
I don't know if it's a rock or what. Just around 6 o'clock, there's something
Okay, well, go
right
ahead.
here that I'm
Finish
not
on
sure what that
Yeah,
is. Yeah. I think it might be.
The one on the bottom, Steve?
Yeah, I think it's a rock. It was just the shadow was giving. I was looking at this thing here.
I see. Okay.
Oh.
Just
Yeah,
it was
keep
giving a
going.
weird shadow. Okay, we can keep going.
Roger. Pull wide.
So how are these rare metals, how are they formed?
Well, they precipitate
Jeez.
out of the water. Yeah, especially the more water mobile elements, which don't quiz me on that. My hydrogeochemistry teacher would be mad at me, but yeah, the more water mobile elements will travel through the water column, and it will eventually precipitate, or it precipitates over time onto the rock.
I'll hold off here for a minute.
So why do we
Roger.
see that there's more metals or more precious or rare metals than we do others, like
Yeah,
iron
wow.
and stuff like that?
Well, like I just said, more water mobile elements are in the water. They're mobile, so they're, you know, and some are more prone to like precipitate and, you know, to dissolve into the water and then precipitate later. Yeah, every element has its own unique characteristics as a function of, you know, their proton and their neutrons and etc.
Yeah. Yeah, I think it's broadcast up. Oh, sorry.
Not much on this side.
Roger that.
Can we zoom on this anemone?
Yeah, sure thing. I'll give you a wee bit of a tug there, Gibson.
Yeah,
Oh,
Roger, I'll
oh.
come down a little bit. There's the tug. I turned auto off.
Okay. Go ahead and push on in. We're just going to be a bit far. It's all good. Roger.
Oh, it's actually not an anemone, it's a corallimorpharian.
That's not an anemone?
How's
Anemone?
No, it's not an anemone,
Wow.
technically. It's a corallimorpharian.
Huh.
And
Interesting.
they're closely related,
Good,
but they're
turn back
different.
on the auto heading and head back to the same.
Corallimorpharians typically have a very broad oral surface, so you can see it's not as closed up as an anemone would be. But also corallimorpharians have knobby tentacles typically, and the defining characteristic is that their internal anatomy is a little bit different. The arrangement of their mesenteries, which are like small tissues that separate parts of their body, are different from a lot of other anemone species.
Wow, there's an overhang.
Oh, there's what I was talking about earlier, actually, down in the lounge. Is a lot of times on these vertical surfaces, you find these really high-density patches of a golden coral.
Good, and push on
These
in
white
there,
corals
please.
here, they're a type of golden coral, Chrysogorgia called Rumelagorgia militaris.
Thank you.
And they can grow in high densities. There's also some regular Chrysogorgia bottle brushes here, as well as I think I saw an irrdigorgia somewhere. But this is pretty typical for this this genus and species, Rumelagorgia. They really like these vertical hangs
Go ahead and come wide there, please.
That's a great shot. Yeah.
All right, I'll start ascending.
Roger.
No irrdigorgia, just the Chrysogorgia. Maybe 2 species of Chrysogorgia plus Rumelagorgia.
Lynette, would you mind doing a DPL reset, please?
It's a big feature.
Yeah, nice boulder.
Oh, is that a
Thank you.
hemi? No. Is that a hemicorallium up there?
Yeah, can we take a closer look at this?
Sure.
And is that another corallimorpharian? Or is that the same one?
I think that one might be an anemone.
Nice.
Go ahead and start your zoom in, please.
The tentacles look a little bit more sharp pointed.
Oh, Chrysogorgia.
So we've got here,
Oh,
ahead and try
we've
to
got
get
2
tighter.
corals. We've got heteropathes right here on the bottom. We've got a Chrysogorgia species.
Hey.
It's a biflobellic colony.
Go ahead and come wide.
It is
Sure.
between 2 species right now, but I would say it's either Chrysogorgia chryseus or stellata. They're, in my opinion, very difficult to tell apart. So we'll just go either of those and look at it back on the
And
beach.
we're back.
Steve, I remember you telling us before, but could you remind me what biflobellic means again?
Yeah, it just means that when the colony initially branches, one branch will form one fan and one branch will form another fan.
Oh.
And they kind of land, they pancake.
Gotcha. Thank you.
Yeah.
To people in chat, sorry for being away for so long. We were having some technical difficulties. Okay, person in chat, you're right. It is. It's a real thing. And that was what that fish was. You're right. I will not be saying that fish's name out loud because it is deemed inappropriate, but just for that person in chat, you are correct.
Well, it's a common name.
That is a common name.
I told you it was ugly.
Atlanta heading 090. You want some?
So Brendan, someone in chat wants to know, could this be a paleo shoreline?
No. I don't think so.
So what is a paleo shoreline for those of us at home that don't know?
I'm assuming they mean, well, you get certain features on a shoreline, certain sedimentary features because of wave action and stuff, and certain deposits are created, and they're not volcanic. They're like sandstone.
I have
So
a small request. Just below you. Just came out
I
of
think
screen.
that's what they mean.
There was a red something.
Sure.
Can
Yeah,
you go
we
back
have a
a
brief
second?
second.
Partial zoom would be
I didn't see any sedimentary features that would make me think that,
about
or just
here.
features in general.
Oh, Raj. Okay.
Thank you, person in chat.
And Gibson, I'll have you winch up in a minute there. Go ahead and push on in. Oh, sorry.
Oh, it's a rock pen.
You want to turn the perch on there too, please?
It looks. It's always tough with these things when they look like this. It could be a Panaculid, or it could
Got a
also
slight
be
more zoom there, please.
Yep.
in the Untitled family.
There we
It's
go.
always tough to tell.
Thank you.
This looks more Untitled to me.
All right,
Polyps
Steve,
are
I'm going to
coming
have to
right
go.
off. Yep, you can
Go
go
away,
whenever
please.
you're ready.
Yep.
But the polyps are coming right off that center axis. Some of the most poorly known sea pens in the world are rock pens.
start
tally.
coming up a bit fast there, Gibson. See the target and sonar there?
Yep, roger that.
It's getting dark. This one right here.
Another bottle brush Chrysogorgia, actually a couple. These are geniculata species.
Oh,
There's a really nice corallimorpharian there. You can see
There's
the
some
white
cool
knobby
stuff.
tentacles.
Oh.
Really more high density Rumelagorgia.
Jason.
For a little while ago, we got a question in the chat. What is the range and endurance of the EV Nautilus? Well, the EV Nautilus, for reference, started out in the Black Sea, all the way in Eastern Europe. It was a Eastern German ship originally, and has made its way over to the Pacific, so a pretty long way.
Bunch of tracks here.
And the endurance, it's a pretty strong ship.
Yeah,
I'm
we
not sure
can
the exact
keep the moves in. We just
whatever,
gotta get over this feature.
the exact strength of it, or whatever units of measurement we could track that on, but it's a pretty strong ship.
I want to say endurance is more like
Actually,
how long
sorry,
it lasts, I
go
suppose,
180 on that and
you know,
say,
during an
we'll
expedition.
wait for a swing.
Oh,
How many days they can
Yeah.
spend at sea, yeah.
Right.
I think
Gibson,
it's,
I'm gonna
we
have
can
you hold
spend,
up a little bit fast.
well, our expeditions are
Roger.
only about
Coming up.
1 or 2 months long, correct?
Yeah, well, it's always depends on what activities are going
I'm gonna have
on.
you haul me up if you
There we go. We're
But
over.
endurance on most ships is food related.
almost.
Almost.
How much can you store?
That was a tall one.
I think
Yeah.
I think it's somewhere around 30 to 40 days here.
Oh, some more Rumelagorgia.
Didn't you ever play Fallout, Matt? Endurance, come on.
They're gonna
I
keep coming up.
have spent way
Roger.
too much time on that game.
You don't know what endurance means.
Well, they were all hours on Fallout 76, so forgive me.
Can we look at this rock? It's very odd shaped, long.
Sorry, Steve. Actually, we won't be able to look at that rock right now.
Okay.
We're making sure we don't slam Atalanta in the cliff.
It's probably important.
Yeah,
Kind
but-
of looks like a bathtub,
I'll let you know.
which
when
is an
we're-
actual thing.
Let's. That's the most important bit.
The lobate flow can break down near the lava tube and create a bathtub.
The Batman of the sea.
Batman. What? Bathtub.
Oh, you thought, okay.
Bathtub.
Answer the question, chat. Sorry if you already got this question. Don't worry about it. What causes these pockmarked surface texture on the rock formations in this area? Brandon, I think you said erosion before? Yes?
Well, we don't get a lot of erosion.
Ah. So how are these pockmarked formations formed?
Oh, this looks weird. Huh. That looked like a really weird feature. And it's sort of, now it's in this like thin bedded sheet flow, I dare say. So the pock marks, the crust will break off eventually, I think. Just because, you know, either internal fractures eventually get larger and stuff like that. And that crust will break off and go away. And you have pock marks, I think.
We do have an update on the eel. It was actually different than any of us thought from our fish specialist, Ken Sulak on the beach. And he was saying that it's actually a type of sawtooth eel, which are a little bit different than snipe eels. They have very long jaws that kind of curve up and down. but they're very good hunters apparently, trying to detect bioluminescence usually.
Spawn until you die.
Interesting. So they have a use for their eyes, and where they can't see normally. Right. I mean, they all have eyes, so there must be something, right, going on?
Yeah. Yeah, they might just not use the visible spectrum like we do. That's okay.
It's okay, fishy.
They won't see me, that's for sure.
I don't get it. Because you're sitting in the van, or?
Both.
Okay.
I wouldn't want to walk into one of those in a dark corner of a seamount or something.
I mean, they're like that big. They're not huge.
Hey, you know what?
You never know. They can get
Have you seen some of those animals in the deep sea with the teeth and jaws they have?
You're not wrong.
Exactly.
I'm sure, but it's like most wild animals. They're more scared of you than you're scared of them.
I don't think they could see you.
The thing
So,
about
you
the
gonna
deep
test
sea
that
is
there,
there's a tendency for a lot of organisms like the squat lobsters. You know, they attack first and then sense later.
Fair enough.
Oh.
Because usually you don't know, like, you don't know or really care what your meal is, you know, if you get a meal
Sorry,
once
can you
or
repeat
twice
that
per year,
instruction,
you know?
Jess?
Yeah.
Oh,
So
goodness.
might as well just,
Yeah,
you know,
roger.
get your prey first and ask questions later. And that's kind of the logic behind like anglerfishes too, right? They have big jaws to capture pretty much whatever comes into their path. The same thing with ambush predators. Like if you notice that tripod fish typically will orient the upslope because the currents are typically downslope. So when the food, their prey, is drifting downslope, they can just you know, jump up and grab, you know, something. A very passive way of being an ambush predator.
Trying for their food.
Yeah, or letting their food come to them.
Nice.
All right, we have some questions for our ROV people up front. Where did it go? How hard is it to drive/conduct these kind of ROVs?
Sorry, one moment.
Of course.
Excellent. It's a lot of spatial awareness, right? So we're always monitoring what certain things are doing. So the ship's doing what, how much layback we have. So it's a lot of fun. Sometimes it's challenging, but definitely a good time. What do you think, Gibson?
I think there is like a sensory overload when you sit in the Herc chair. There is like, and not only are you piloting Hercules, which is, as Jess has said, it's an amazing like exercise in spatial awareness because you're got this 3D space you're basically levitating through and you're rotating in all directions. But then you have the ship moving above you and Atalanta rotating and bouncing around you. And then you have the science objectives and you need to make sure you stop and get the zooms and the samples that you need while everything's moving over this uneven seafloor, trying to run into cliffs or stir up the bottom. It's amazing. It's a learned, takes a lot of practice, I can imagine, because my first shot at it was, I certainly got a taste of how much I have to learn. So from my perspective, whoever asked that question, it is challenging, but it takes practice. You can get good at it.
So do you guys, would you guys say you prefer flying and piloting ROVs or more working on them and launching them and recovering them?
Ooh.
They're so different, but so fun.
What is that?
I think they complement each other.
Big,
That's not.
not organic.
Well, it might be a piece of wood.
It's like a branch.
Looks like a stick.
It does
like a
look like
piece
a
of wood,
stick,
yeah.
yeah.
Woodfall.
Woodfall.
Just
Yeah.
one.
Can
Yeah,
you-
I'm not sure. Everything, when we're working on the vehicles, that's a lot of fun. A lot of troubleshooting. There's troubleshooting going on right now. And when we're flying, it's yeah, I mean, I'm partial to, I love flying. It makes you feel like, I don't know, you're seeing something new every time. So that's, I don't know, a big treat.
And can these flying skills, quote unquote, can these translate well into other vehicles, or is it just specifically ROV centric?
I think that the skills for ROVs are very ROV centric. You know, you have tether, clumps, weights, stuff like that. We have other folks that pilot HOVs, so human operated vehicles. And in that case, it's different skill set, or same and also different. You know, you don't have a tether, you don't have to, but you have humans inside. When it comes to like helicopters, I don't know. I'd have to learn a whole new system. I don't think you guys want me to fly a helicopter right off the bat, you know what I mean? And I'm probably really bad at Mario Kart, so
To be determined.
That's
to be
that's
determined, but I'm pretty sure I'm bad.
If anyone's racing Drew, they're going to look bad. That's all I'm going to say.
See that
I'm
good?
sitting back here, and that's your comment?
Brandon is our resident Mario
It's a sore
Kart champ.
subject.
Brandon, are you trying to say you're good, or? Not
I'm
really.
the best on the ship.
Okay, well, I came in 9th place, so I don't really know what you're talking about.
All right, dude.
Yes, please.
Don't gotta flex at us like that. It's okay. I'm apparently the worst Super Mario Kart player on the ship as well, as I sit in a comfortable 10th position constantly.
As I sit in the 12th position usually.
And yet she still beats me.
I don't think so. I think
I
you beat
think
me
we're
last
pretty neck and neck for
Okay.
last.
Kukui, I think it's golf rules. I think lower the score is better.
Okay, that's 090 there.
Roger.
If you personally could name a new species of fish, what would you choose?
Sea bream.
Yep.
That one's a Cophobolumnon, almost certainly.
Cuffle what?
Cophobolumnon.
Cuffle balloon. I can't even say
Bless
that.
you.
Go ahead and start using, please.
Of course.
I had to go to 27 years of school to say that.
Cuffle balloon.
Stand by.
Have
And
some.
it's every syllable is worth it.
Cophobolumnon. Yep.
Balumnun.
All right.
I feel like I'm still
Kukui.
saying it wrong. I'm sorry.
Steve, are those dark bits? Is that
All right, go
food?
ahead and push on in some more.
That's the what we call the GV, gastrovascular cavity.
Okay.
The guts, basically. They don't have intestines. It's one cavity
All right, full life,
with a
please.
two-way opening. Food goes in, food goes out the same hole.
All right, gonna start
And
coming up again.
occasionally, if they do have reproductive organs, they'll usually be at the base of the polyp. And as they produce more gametes, they'll take up more of the body wall of the polyp. But I didn't see that there.
Gotcha.
We saw some umbellula the other day, a little bit shallower, that had very large yolky egg masses at their base. Oh, there's something over there, a lobster.
Oh, what is that?
Oh, yeah.
Wow.
Sorry, guys. We actually have another boulder coming up, so we will not be able to look at that. Please start coming up on the winch.
Roger that.
That was a
I have a small picture of it, if you want.
yeah, no,
And if you don't mind maintaining
it?
a 15-meter delta.
yeah,
please.
that's a
Roger.
lobster.
Wow.
One of the, let's find the name of that thing.
Yes, Jason.
Delta's at 1.5.
Roger.
What depths are we operating at, and what are the depth limits for the ROV
Like
vehicles?
kilos?
Currently, we're just under 2,000 meters. We're at about 1,980 meters deep. And the ROV depths, ROV team in front, feel free to correct me if I'm wrong, but I believe Hercules is 4,000 meters, and then Atalanta is 6,000 meters.
Gotcha.
The reason being is Hercules has foam on top of it that keep it buoyant, and if we go any deeper than 4,000 meters, that foam starts to cave in on itself because of the pressure.
It's an interesting looking lobster. Reminds me of a horseshoe crab, kind of.
It really does.
My favorite, you know, I do have a favorite sea creature, and it's actually one of the other deep sea blind lobsters that we see. Just, these things look so sci-fi. Let me see if I can find it.
But can you eat them?
If you're brave enough.
You can eat just about anything.
Oh, is it?
You might
is
not
it,
be feeling too well on the other side of it.
Is it safe to eat?
It's safe to eat it, but not digest it, I would say.
Good one. Asking the real questions.
Essential questions. Another battle-breast Chrysogorgia.
So, Steve, would you say this is your favorite
Acanthonus.
invertebrate? This kind of lobster that you? Oh my lord!
Wow.
Does it make its own like burrow too?
No, they usually don't. They don't have to be in burrows,
Okay.
but they have these mantis-like jaws that are just gnarly. Acanthonus. We see them in the Atlantic quite often.
Steve, is that your favorite invertebrate, or do you have any more?
It's one of my favorites. It's pretty awesome for arthropods. For a crustacean.
Ooh, an irrdigorgia.
Yeah. Irrdigorgia, Magnispiralis.
Okay, wait, what was your favorite coral again?
Glass sponge.
I don't like to say that. I don't want to insult them.
Okay. all of them. All of them.
The
Yeah.
non-sentient beings of coral being insulted.
Hey,
Who knows? It could be
that's
sentient.
insulting in and of itself.
Sorry,
Very
coral.
sentient.
Very sensitive coral.
I mean, I have a favorite rock.
When the corals rise up and take over the world, you'll be the first one.
I'm oh no.
Brandon, what's your favorite rock?
Schist.
Shit. What'd you say to me?
Yeah, exactly. Schist.
Schist rock.
Is that a
I'm
sea
not
star? Is
gonna try
that
to
a
say that.
crustacean over there?
It's a Brisingid sea star.
Crustacean sea-
Yep.
Oh my goodness, I need to get my eyes checked.
Nah.
And then we've got a Chrysogorgia bottle brush. Very nice Chrysogorgia geniculata.
We got two of them, it looks like.
Yeah, maybe two. The other one's very small.
Yeah, another-
Both are occupied by squat lobsters.
This terrain is nuts. It's cool.
Actually, I think it's rocks. that was a stupid joke. I'm sorry.
It's a lot of nuts piled together and then the rocks formed on top of it.
That was a schist joke.
Be very, very careful with that wording there, Brandon.
The bubble cam looks really cool too.
Yeah, it does. It looks like steps, very steep steps.
Yeah.
It's
Yes.
probably going to get steeper.
Sweet.
Yeah.
These are very gorgeous pillow basalts we're seeing.
What's your favorite kind of flow?
Pillow basalts.
Really?
Yeah.
Why
It is.
Why?
It makes up most of the Earth's surface, which is a cool fact.
That's a
Ah.
cool fact.
And look at them.
They rock.
Such low-hanging
Use that
fruit,
one, yeah.
and we hit it every single time without fail.
We take these puns for granted.
Oh,
Ah, that's like the second. That's the second one that we always do.
Has YouTube ever shut down a channel for bad jokes?
For offensive ones,
I
maybe.
never really get these rock
Are you
puns
offended?
'cause
Is it time for dinner?
Go ahead
I
and
don't get
fish
these
in
rock
and
puns.
I'm
I
not
think my head
offended.
might be too
Okay.
dense.
I hear this all the time.
Does I have some?
Wow, that's really cool.
Ooh.
Another Euplectillid glass sponge.
Glass
Go ahead and come mine, please.
Sure.
What's on top of it?
That's
Egg
crinoid.
crinoid.
fudge and a crinoid?
Yep. And a bolosoma sponge in the background too.
Oh.
I need to brush up on my crinoids. Not very good at them, identifying them.
Thanks, Annette.
The stalked ones I'm doing okay.
Have we seen a lot of floating crinoids?
Yeah,
Whenever they get knocked off of a coral.
arguably more than coral.
Those are free
There's a
falling
fish.
crinoids.
Here.
Sorry?
There's fish on the
Oh.
right side. Did you see it?
Did you want
Oh
to look
yeah.
at it?
Yeah, maybe.
I don't know if we'll get over there, but
Yes. Okay.
With our peepers.
Good eyes.
Yeah. Wow.
I lost it. Oh, there it is.
Yeah,
Yeah.
that's right there.
Wow, very good eyes.
Might be just a cusk eel.
You want the porch?
sure. Go ahead and start your zoom there, please.
Sure.
There's plenty of rock pens here. There's another rock pen too.
It's half so.
Thank you. Oh, I'll see ya.
They said bye.
So I think that's a synaphobranchid cutthroat eel.
Cut throat.
You want that porch back off?
Oh, yes, please.
So Brandon, somebody in chat wants to know, do you know the geological origins that form Johnston Atoll?
I am aware of the geologic origins.
Please, would you be willing to share said geologic origins?
Yeah, so the basic way of saying it is we think they're around 75 million years old. And it's a mafic basalt for the most part. And then, well, here, let me start over. So it formed from a mantle plume is our best guess. There's sort of these upwelling features that come from the core mantle boundary. And they're hot enough to create melt and to melt through the oceanic crust, which normally isn't possible with upper mantle material. And that creates all the basalt. And eventually it becomes an island, and then that island has a coral reef build onto it, because that's what corals like. They like that shallow water, or at least coral reef corals do. And you know, as the island has slowly subsided until it became an atoll, which is just, you know, a low relief island with coral reefs surrounding it.
Hey, Lynette, after this move, let's hold position for about 3 minutes or so and just let Atalanta swing
This rock looks like a boat.
before we get into those really
Boat
tight
rock.
contours. Thank you.
What kind of
That's some
boat?
weird,
Very angular rocks
Yeah.
right here.
Yeah.
especially long.
Is that a rock in the center there below the lasers, or is that a sponge rubble, or?
It looks like dead sponge.
I don't see what you're looking at.
Right here.
Oh yeah,
yeah.
I don't know.
It looks
We
very
can take a look.
It's so pointy, yeah.
Go ahead and start a zoom there, please.
Sure.
Ooh,
It's a
what?
half so.
Sure thing.
Another, yeah,
Wood?
I don't know. It's playing with my eyes.
Yeah.
Tough to tell.
All right.
It's
Got
very
a little more
thick.
zoom.
Nah, I had to go
Alright.
full leg. For this feature here, we have a clip.
Answer the question of how long is this expedition? This expedition is going to be about or at least this dive is about 20 hours. I assume that's what you meant by that. So we launched at about 9 o'clock, 8,
It was 10 something.
10 something in the morning. And then 20 hours after that, you can do that math, and that will be our recover time.
6 a.m.
6 a.m. will be our recovery time.
Rumelagorgia. Some
So tune in
particles
then.
there.
But the expedition's another like 6-ish days, maybe 3 more dives after this one.
Yeah, we got about a week left. The end is in sight. Here's another question from chat. How much of the seafloor is fecal matter and dead things?
Probably a lot of it.
In terms of percentage, or
Yeah. Like, do we know
A yellow urchin like thing?
a percentage on
Tennis
average?
ball urchin.
Yeah.
I would say less than 5%. It seems like a lot because that's all we see. But the basalt is on average about 8 kilometers thick. So
Yeah, if you want to push it on Atalanta.
You know, that would be a really interesting question for Rob, because I think
Yeah.
he might know something about that.
that's true.
Hey Rob, if you're listening from the lounge, how much of the ocean floor is poop,
Bacterial.
bacterial, and other deceased beings?
Ooh, a sea
what a
cucumber.
bright cucumber.
Oh wow.
Yeah.
I love the contrast, that purple to like
A
the
lot of
gray
carbon
rock.
is really efficiently recycled in the deep ocean. You know, large reservoirs of carbon, like dead carcasses of things, are fairly readily consumed by the locals in the deep sea.
The locals.
It's pretty angular stuff here.
Yeah.
Looks pretty fresh, maybe.
Yeah, be nice to sample.
So,
That's true.
is it impossible to try with the arm again?
It's not impossible. It's just it won't fit around the jaw width opening. If you can see my hands, and the
Oh,
intro
yeah.
end's like that big.
Okay,
So it doesn't really fit around most samples that we would want.
got it.
Yeah.
Is that the same jaw you were carrying the fossil in?
Yes, that's correct.
Gotcha.
Yeah, so it's positioning it and then getting a sample in. Like some samples are flatter, right? But
Right.
getting that sample to actually sit in the jaw is an hour-long chore. Lynette, would you
Some
mind doing
nice
a video
Argus
of this,
shots.
please?
Well, they were until you said that and I nosedived. Yeah, it's neat.
Looks like we're settling out nicely. Maybe another 2 minutes or so. And as we kind of get into the thick contours or the really narrow contours, I'll just, if you don't mind doing like the 20 meter, then we'll wait. And then 20 meter wait, especially because they're pretty, this isn't even the tight contours yet. Thank you.
Jess, how's the zoom on the Atalanta cam? Can we try?
Oh yeah, totally.
Danny, you want to try pushing in there?
I would love to.
Let me swing a little left for you.
Let's see what we got here.
Nice.
Ooh.
Look at that.
Uh-oh.
Have a
Oh, is this
teleporter.
columnar basalt? Oh, wow.
Wow.
Nice.
I've been waiting this whole
Highlight.
expedition to see this.
Wow, this is gorgeous.
Maybe
That's
the biology
cool.
likes columnar basalt.
I keep asking Brandon, is this columnar basalt?
Keeps hiding it from us.
Yeah, this is definitely columnar basalt.
Cool.
call it columnar basalt.
Do you guys have some nice shots in the
Okay.
cam there for you?
Yeah, we got some nice StelCam
Do you want me to put
angle
the
going
cam
on.
up for you?
It's a
No, it's okay.
Paragorgia too,
It's okay.
bubblegum
Oh,
coral.
wow.
Wow, that's really
Wow.
Wow. Kikui, that was the best joke of the watch.
Thank you.
I
cool.
missed
Oh
it.
my
very much.
goodness, look
Whoa.
at the Atlanta
It's like an
cam.
amphitheater.
Wow. Here, I'll zoom out for you.
Yeah,
Yowzers, that's cool. I'll take some without the laser on too.
It's like
Yeah,
the
I'm just gonna
nosebleed
check
section.
out still.
Yeah.
Madison Square Garden.
There you go. That's pretty decent on the DSC.
Wow, cool. Well, I'm kind of glad I stayed for this extra
Man.
shift.
Yeah.
Sponge rubble too.
That's cool.
Hey, can we
Okay.
put a target down here and call it columnar basalt? Thanks, Nev.
No problem.
Very cool.
Oh, there's a sparse branching bamboo too. Wow. Yeah, this is nice.
For once, the geology really is trumping the biology here. No offense, Steve.
Them's fighting words.
That's fighting words, yeah. Very cool.
So there's that sponge rubble on the bottom right? That like,
Yeah, there's a big,
huge.
maybe a Phoreid or a Tritopleura sponge, possibly. Yep.
If you want to push in more on the
You said that was a
Atlantic here.
Paragorgia,
Sure.
right? The yellow
Yeah,
one.
that's a Paragorgia with zoanthids. Yep.
whoa,
Yeah.
cool.
This looks fresh. Yeah.
Nice shot, Dan.
Look at
Freshish,
that.
I guess.
There we go.
Oh,
We had it for a second.
I'll wait for her to calm down.
They don't make
Yeah,
Dramamine
even if
strong
you
enough
set
and
it
strong
back,
enough a
yeah,
dose
okay.
to tolerate that view for too long.
Wow.
We could look at it all day. I wouldn't mind.
Yeah. Can we take some Argus grabs here?
Yep.
Or Atalanta grabs?
this bird.
If we can go to the middle of the feature too,
Yeah,
curious what it looks like at the end.
sure thing. We might not have the scope right now, but we will get there. And in the meantime, we can enjoy some nice Atalanta view.
Oh,
Beautiful.
Just kidding.
sorry.
You're all good.
I'm willing to heave to stop. I'm trying my best.
All good.
Looks like a branch bamboo out there.
I've recently learned that even my best attempts at active heave comp on the winch are
Utile,
to
right?
no avail.
Totally, yeah.
Gibson, is it just like an entirely different skill set piloting Atalanta versus her?
Oh yeah.
With the winch and everything?
Yeah, I mean,
Yeah.
Atlanta, there's a lot less sensory overload for sure.
Brandon, we'll go back over there. We'll just kind of mosey around here while we wait for that ship move to be felt.
Got it.
I remember years ago watching
That's a cool shot.
Nautilus live and always wondering why one camera was always kind of, so much more erratic than the other, and it totally makes sense now.
It's because Gibson's up here pulling the levers
Yeah.
like this.
Untrained pilot versus trained pilot.
No, I didn't mean that.
You want to go ahead and push in partial on some of these really cool columns?
Sure. It's the ocean's fault. It's not Gibson.
No, of course.
But when it's a good shot, then it's my
Right.
fault. Yeah.
I can prove it I can prove it to the viewers at home. Sat feed 3.
Very cool.
There you go.
So the
That's awesome.
That is awesome.
the way these features form underwater is not well understood actually,
Oh,
at least to my knowledge. But on subaerially, so above water, they form as the magma. It's always basalt too. Basalt likes to form like this on the surface. When the magma goes over like an overhang or a cliff, and as it's flowing down, it cools and
Go ahead and
creates
come
these
right
awesome
there.
columns.
Thank you.
So it's more about how it's
stalks granite.
cooling rather than how it's flowing, or?
Both.
Gibson, you're wide now.
thank you.
Yeah.
Yeah, at least on land. But not sure how they form underneath the water because it quenches. It cools down, it crystallizes so quickly. It's thought that they wouldn't be able to form these columns, but as you can see, they do form. So
It's magical.
That's
I'm
beautiful.
going to have to ask
Yeah.
Kevin or Rob, who picked waypoint 2?
Is that where we are currently?
Yeah.
Yeah, just about, because that's where
A little
that's
past.
where we found this nice
Is
feature.
what you call a bullseye?
Bonus watch
Right
bumps
on.
to them.
Look at that nice like cliff to the bottom side too.
Oh, wow.
Very cool.
What a cool spot this would be to dive if we weren't squished to mush.
I wonder if the orientation has anything important to say about,
I would wager it does, yeah.
so it's
Come
kind
on
of like
down if you want to
southeast
go
to the
Roger.
northwest
Bring us down to
you
1.5
know,
here.
along the axis of the columns.
And while we're waiting here, feel free to push on in there, please.
Sure.
I love how the
Nice
serpent
sea
stars hang
serpent.
out in the center and just kind of like show themselves off.
Yeah.
Yeah, they really know the prime real estate.
They would be awesome sunbathers if they could.
They would be. And do they do that to catch? No. What do they feed on again, serpent stars?
They're planktivorous, so they're probably capturing you know, bits of food, whether it's probably marine snow out of the water column.
Sure.
They have sticky tube feet on their branch tips. Sometimes you'll see the tentacles streaming
Roger
off
Right.
the colony.
Thank you.
So that widespread is beneficial for their eating.
Yeah. They might also acquire some nutrition from material cleaning off the coral. You know, as they move their arms,
Yeah.
they might clear off some mucus that might be, you know, food for them.
So cool.
Let me get this straight. Some sea stars eat coral and some stars eat, but they hang out on the coral and eat other stuff.
Yeah, I mean, I have not yet seen an ophiuroid or brittle star that actually eats coral. It's mostly the asteroids, the true sea stars that are the predators, but also urchins to a lesser degree.
Gotcha.
And the crinoids are kind of you know, somewhere in the middle. They don't really eat the corals, but usually there's tissue loss associated when they latch on to the colony with their cirri.
Awesome. So what does this flow pattern tell us here, Brendan? It looks like this is the jointing that the Argus cam has really good
Yeah.
view of it. Nope.
Yeah, I mean, the columns are, you call it jointing, separated by fractures. It's interesting, it kind of has two orientations, one like that, one like that.
Yeah.
Could be two different flows coming together.
Hmm.
Would have been spectacular to watch this form.
Yeah. If you're here 70 or, you know, however many years ago, 100 million.
Well, when you say it like that.
I'll take the lasers off for a nice shot too.
I don't know, maybe in the metaverse we can go back 100 million years.
Oh my gosh.
Well, you can watch volcanic features form in the modern day.
All right, I'm gonna
Without-
start coming up a bit.
Without the metaverse?
Yeah,
Oh.
at mid-ocean ridges, which we're not at a mid-ocean ridge, but
All right, Gibson, would you mind acquiring a 090 heading again?
090, coming around. Can you go wide, Dan?
You bet.
Thank you.
Thank you.
yeah, some people call it columnar jointing as well, instead of columnar basalt. Yeah, awesome
And then
feature.
once we get stable, I'll have you do a DVL reset
Oh,
again.
branching from node.
Thank you.
Yeah, I'm not sure what that is. It does look like a primnoid. There's umbellapathes too. And actually this stalk in the background, that's actually a stoloniferan. If we do have time, can we take a peek on that?
Sure thing. Go ahead and start your zoom there, please.
Sure.
Yep.
So,
So this is
Whoa,
this
that's
is
cool.
actually an imposter. It's a
Imposter.
sponge
Dun,
stalk
dun, dun.
that's colonized by stoloniferous octocorals, and we've sampled this on like previous expeditions. But it's always curious because sometimes they can occupy different morphology stalks, and it looks like they might be making it, but they're not. They're taking advantage of someone else's hard work.
Oh.
wow.
Wow.
Go ahead and push on in a bit more.
Sure.
They're
So they
opportunistic.
stole the sponge stock? essentially?
Yeah.
Pulls
I
it.
mean, well,
Thank you.
if the sponge is dead, did they really steal it, or are they more squatting? Yeah,
Full white, please.
Squatting sponge.
tough to tell. Maybe opportunists is a better way to say it.
Okay.
They're opportunistic.
What is that
Bamboo
by
coral?
that? Oh, bamboo coral?
Yep.
Bamboo.
Look at all this coral.
Old, what's that called again? Base, coral base.
Yeah, there's some pretty good coral diversity here. We're getting there.
Oh,
Maybe
is that a
we're
hemicrowd
finally
over there?
getting up and over the ridge that was waypoint 3-4 that was blocking us. So maybe there's a
Ooh, nice shot.
nice
Jess.
current accelerated topographic flows around this.
Oh, that's nice. Right.
Topographic
on the
accelerated
axis.
current flows
Wow,
around this.
almost. It's too
Topographic
bent. They really get
accelerated.
one.
No, we can
Current flow.
No, this is a waste of everyone else's time, though.
No,
Go
this
ahead
is
and push
awesome.
on in, please.
Oh,
This is
yeah.
super
Ah,
cool.
yeah.
Nice job.
It's like looking into the metaverse.
Wow.
Just like looking into the metaverse, yeah. Go ahead and come away, please.
Sure.
Thank you.
Wow.
That's
Down.
Fancy flying.
irrdigorgia magnispiralis.
It's like someone just made a house here. All these bricks.
Yeah.
You want to push it on Atlantic?
Is
in
it
there,
brittle,
please?
you think?
I don't think so.
Just get a little love tap, see what happens. That was a joke. Please don't do
So
that.
I'm just going to see how far
Yeah,
this
go for
actually
it.
goes. Oh, wow, that goes very far. Okay.
Oh.
Does anyone want to lose their dinner tonight?
Pulls in.
I can help. All right,
That's
half
such
before
a
you
cool
get
shot. Yeah, right.
Yeah, that is.
Cool shot.
Joke's on you, I
Well done,
haven't had
Gibson.
dinner yet.
Oh, I did.
I got
Well,
nothing to lose.
A sponge.
Oh,
Walteria
what's going on there?
Go ahead and push on in there, please.
Sure.
with Brisingid, actually. Or no, yeah, with a Brisingid
It's half set.
Thank you.
and Unidopsid, or
Wow.
I'm going to push it
What
on
are
the
they
little
called?
crab guy thing.
Yeah, absolutely.
Wow.
Whoa, that's cool.
Look at that.
Hello there.
look at him go.
Look at his little, oh, no, not little, never mind.
that's yeah, there's
Full white, please.
a name
And
for
then
this
You want to go ahead and push another move there, please?
squat lobster.
That is a bright lobster. Jeez.
Yes, please.
Oh.
It almost looks like a bridge.
Calatheid. How to get the right family.
So Gibson, you're saying you've been waiting the whole expedition for all these columnar
That's
basalts?
right, I have been. And now I'm like dreading a dinner break right now.
It
It's
would really,
cool we got to see one on our watch, though.
I know.
Yeah.
I'm pretty
Yeah.
stoked about this.
That's awesome.
it's definitely climbable.
Yeah, no kidding.
Underwater climbing.
That's all I've been thinking about.
Answering that
You got
question.
a long coral to your left there, Jess.
A long coral to my left.
Yeah,
Bamboo?
I think so. It's hard to tell. There you go.
Oh, good eye.
Thank you.
Didn't even see
Yeah,
that.
it does
wanna
look like
go.
bamboo.
Wow.
Oh, oh,
Kidding.
oh, you were not kidding.
Yeah, it's pretty long.
Oh,
That's a
okay.
long
All right,
boy.
dinner break.
Oh my god.
And with that, I leave you.
Beautiful.
Oh my goodness.
How? Oh,
This is like a clown
Bamboo,
handkerchief.
coral. Look at that.
oh my gosh.
Pull that out of my hat here. Go ahead and push on in on the polyps there,
You
please.
bet, you bet. Wow. Look at that.
Very cool.
Wow.
Nice. Jeez.
Good.
Nice flying. Look at that angle.
Beautiful shots.
Straight down the barrel, yeah.
Great eye too.
Good. Good flying.
Thank you, guys. All right, we'll go look at that wall behind it now.
Alrighty.
Wow, that's a lot of Rumelagorgia. I think that's Rumelagorgia.
Our coral guy just went to go get dinner, so
Full wide on Analat in there.
Rumelagorgia,
That's
right?
Rumelagorgia, and then probably Norella, and that one on the bottom may be a bamboo, or it may be another primnoid.
All right, I'll go back to where I need to go.
And some more bathypathes too.
Yep, and there's a bathypathes, several of them.
Is there a stauropathes in there somewhere? Oh.
We have seen stauropathes. I don't think we've seen them yet on this dive.
Coming back to you now. That's funny. That's fun.
is there a simulation where you can try piloting Hercules? I don't believe so. I think piloting Hercules is only a in real life, in the van experience on the EV Nautilus. But if you'd like to take a crack at piloting Hercules or Atalanta and like to work as an intern here, you can always go to nautiluslive.org, click our Join tab, and you can always have a shot of signing up to be an intern on our ROV team. And then you can pilot one of these miraculous ROVs.
All right, Jess is going to be handing over for a bit. Oh, we have the craft maybe back. Cool.
Hey!
Ooh.
And if you want to talk to an ROV intern, the gentleman who just went to dinner at Gibson, you could ask him some question when he gets back.
That's true.
All about getting into it. What a great idea, though, to have an ROV simulator.
Maybe
that'd
that's
be,
something in the works.
that'd be very something very cool.
So here's another question from chat. For our back row, how many, if any, species are observed on these dives are specific to this region or endemic?
Depends on how broadly you define region.
Johnston Atoll specifically.
I don't think we've seen anything that's specifically endemic to Johnston Atoll. There is a biogeographic province that starts just north of the equator and extends, including Johnston, up through the Hawaiian Islands, Papahanaumokuakea, and up into the most southern end of the Phoenix Islands. And we have seen a lot of things that are endemic to that biogeographic province overall.
So Kevin, we've already asked Brandon what he thought of this. What do you think of these kind of formations?
They're really cool looking. So you got large, slow cooling magma. It's kind of cooling from the sides, interestingly. It's getting these lateral columnar joints forming. It's hard to tell if this is one massive flow. It's probably too big for being one massive flow. Probably just stacks of similar styles. But it's quite interesting it's a ridge because it's basically steep on both sides. So I don't know if it's collapsed. But if you look on the left side, you can kind of see it's a little more pillowy. It's probably the top or the sides of the flow. And I can't tell if the other side is a collapse feature or if it's just a really weird structure. But it is cool.
Yay!
Yay! We got our hand back.
Welcome back, Han.
All right, now let's grab one of these columns.
Yeah, because can we test the manipulator by triggering a Niskin right here?
With the freeze?
Great
No
timing.
way!
Is that the magic school bus?
Maybe.
Oh, I love the magic school bus.
All right, back row. So here we have a sample request.
Yeah, if we could trigger a Niskin right here, this is the first good coral community we've come into, so that would be fantastic.
That sounds great.
I
Right.
feel honored.
Kylie is on the manip. We got a new manip.
We got a new manip. Don't break it again. Got it. I'm nervous. Okay, coming on.
You got this.
I believe in you, Kylie.
Wow.
Let's take a Niskin.
Talk about timing.
I know. Yeah. All right. And pull rack back.
Raj, Raj.
There we go.
Pull wide.
You are.
Okay, triggered.
Hey.
Yay!
Woohoo! Thank you.
Yay! Was that sample bottle number 6 or
Looks.
was
Yes.
number
looks
6?
like
Number
it.
6. Awesome. Thank you.
Yeah. It's great. Good job, guys.
Awesome. I'm going to switch out. Thanks for the fun time, guys.
Bye, Jess.
Thank you.
So question, here's a question for you, Kevin. Why is the columnar basalt horizontal and not vertical? I've seen a lot on land, but they're always vertical.
Yeah, typically you find it vertical because you get the cooling starting on the top, and then it creates the kind of hexagonal fractures that then propagate all the way down. So in this case, it's probably a lava flow that intruded outward and cooled from the sides as well as the top. And so it might have cooled quickly on both sides and propagated inward instead of from the top. Since we're in a marine environment, you can potentially get it coming off the ridge or something like that. But it is unusual to see it like this. There's also always the chance that it formed vertically and then fell downhill as a huge slump.
What are the chances of that happen? Like, that's a pretty low chance of happening, or is it just more?
It's not that low. It's low chance of it being this intact. So I don't think that's the case. If you find just a large chunk of it, it's not impossible. But my guess is these cooled from 3 different angles.
And so it cools like that and makes these sort of step patterns, or does that happen over time?
So that's partly part of erosion too. Top of it probably came off some faces. But yeah, it's going to cool in from the sides over here and then cool that way, propagate in.
So here's another question. Can you explain why the term colony with, why we use the term colony with coral versus just a stick of coral?
Well, they're colonial animals, so each of those individual polyps is technically an individual animal, but they all grow together. They can exchange nutrition and kind of like share resources, but they can also act independently in terms of feeding and opening and closing. So they're a colonial organism.
So are they considered a hive mind, or?
They don't really have a mind. They have the nervous system in those is like a couple cells.
So a hive nervous system.
Yeah. So they mostly just respond independently to environmental stimuli, but they can work together. So say when a sea pen needs to withdraw into the sediment, all its little water organisms, they have a special polyp type that just takes water in and out, will expel all the water, and the colony will retract.
So can coral grow, like if if there's 2 coral next to each other start off and they continue together, are they part of the same coral now, or are they 2 still different?
Depends on the species. So there are some corals that can anastomose together. A lot of them will actually actively fight each other if they grow together. Yeah. So particularly in like reef environments, a lot of corals will have specialized stinging polyps that they can use to defend their local turf.
So can the other coral feel these kind of stinging polyps and these sort of coral smackdown?
It destroys the tissue.
It doesn't matter if they feel it. It's not going to be there.
You can watch it in time lapse. It's rather impressive.
Put some epic music behind it, so WWE commentary.
Yeah.
Let me know when you're ready to start moving it forward again, Dan.
Roger. Let me get stretched out here a bit.
Yep.
See, give me a minute, see what's happening. Take it all in, as it were. Just gonna slide to the south a little, see what that looks like.
Stocked crinoid there?
I'm feeling, looks like it. I'm feeling like we want to slide to the north, go up the point.
Slide to the north. Slide Argus to the north?
Yeah.
Okay.
Argus is on deck. We can slide Adelan to the north.
Well,
that would be
it would both
Argus,
happen.
Yeah. Yeah, you can slide Argus to the north.
Okay, I'll do a 10 meters. Yeah, that would square me up about with where you are now.
Yeah, let's do 20. 10 meters is...
Right into the, and I'll just come up.
Bridge nav, 20 meters due north.
Go.
20 meters due north.
No, my luck will change as soon as we start going up, but... Assuming the nav's correct, I have a kind of a point here, so...
Okay. I'm going to switch your position to USBL.
Roger.
Because we keep losing the DVL, so.
Yeah, right.
You want me to turn the craft off and see if that's the ground fault?
While we're waiting for the ship, give it the subsea milk.
Sure. I mean, it didn't have a ground on it at all today. It was the DVL.
Oh, yeah.
So I don't know if it's the craft or not.
Oh, yeah, 200 Ks.
That's the range that the DVL was. It was 190.
Roger. You can turn the DVL off, not using that.
Okay, Raj, turn the DVL off.
Can you bring your head to the north a little while we're
You got it.
north there? Yeah.
Brandon's back.
So how long has this feature been going like this? For a while, or?
It's been going on for a little bit.
Since the beginning of dinner, I think. Yeah, the ground fault went away.
You can turn it back on. I think Chris needs it for his...
Narbit.
Yeah, well, it doesn't hurt, even if it's unreliable. A couple pings here and there help a lot.
Almost looks like a spiral staircase. And down.
Pretty wild.
Thank you, person from Brazil.
You guys already saw all this stuff, I'm guessing.
Yes. Here's a question for the team. I've been off the grid for the last 10 days. Can you give me a summary of the coolest finds and discoveries that we found in the past 10 days? Well, I mean, first of all, we found a beaked whale skull. That was pretty cool. We found just a little while ago, we found that saw eel, I think it was, right?
Okay, once you get there, you can
I think it was a snipe eel.
step it
Snipe
east.
eel.
Okay.
That
Ship's
was
already there. Do you want to wait for Atalanta a bit, or
No, you can.
Yep.
This is probably the coolest thing we've seen geologically, I think.
bridge nav 15 meters due east?
We've seen some cool, other than that, we've seen some cool pillow basalt flows, of course. We got into some limestone at Aguillo. Got into limestone at Johnston, which is cool to see. We went to this ridge, this really, I guess thin and high relief ridge that could possibly be some kind of tectonic feature, which is really cool to see. Yeah.
We've seen and surveyed some interesting deep sea coral gardens and sampled a few species that we're going to be confirming what they are. Sampled some interesting urchins and sea cucumbers.
Saw a Dumbo octopus.
Oh yeah, a couple.
I get down with that. Yep. Carrier crab. I love the carrier crabs. That's probably my favorite.
The carrier crabs are
He's
pretty
carrying
great.
an anemone on his back.
Sharks.
Oh, yep. There was that
Although
big shark, huh?
We've
6
never
gill?
seen them here on the Delta Janela Benthic Storm Watch. We're the only watch yet to see a shark. Clearly see a shark. And the
Do
other
better,
teams-
Delta Dan.
What's that?
I'm just. Okay.
Do better.
Sorry, I'm in the zone here.
The cutthroat eel, probably.
Oh, we did get
Yeah, that looks like a cutthroat eel.
cutthroat eel.
Follow the eel for a while. Maybe he's going somewhere interesting.
It's a really steep rock face. It's like an 85 degree.
It's a good sense of it there, doesn't it?
Yeah.
Thanks, guys.
Hey, Gibson's back.
I put the bubble cam up on channel 3. It looks sort of looking straight down
Bye,
the
Gally.
ledge there. Right down the cliff.
Back in Columbia Basalt Land.
Yeah, it looks like they look like precariously stacked books. If that makes any sense.
Never heard them described that way, but I suppose.
You can see it, right?
Yeah.
Minecraft.
Minecraft.
That's what I was thinking.
This should be more Terraria, if anything.
Good one.
Thanks. Thanks.
Send that a plug-in, Minecraft Basalt World.
I don't think Minecraft has columnar basalt, funnily enough, but I mean, these are block shaped, so.
Next update.
Crafty work on the craft, Dan. Well done. Was it
You get that dramatic black background there on the big coral. Don't quite have enough wind.
I can come
Is
down a
this
little bit.
Chrysogorgia?
Any other guesses?
That's the only one I have in my repertoire.
Or
That
do you keep
bubblegum
stepping east,
coral.
Dan?
It could
What's
be
that?
bamboo.
You want to keep stepping east?
Can't tell if I can see nodes or
Reach
not.
nav, 1-5, due east.
Can we zoom on Dan?
Sure, go ahead.
5
Wow.
watch bucks for the first person who gets it.
Precious coral. That's it. That's all I got.
It is a bamboo.
Oh.
It's a node branching bamboo coral. I believe we have sampled this one already. It looks very similar to one we sampled on our second or third dive.
And what is that? What is that stock?
That's a sponge. That's something else. I think this one is a J-clade.
Suck me in.
Given how much debris was down at the bottom of this thing, this feature, you know, angular blocks and stuff, I wouldn't figure this would be a hot spot for corals, but apparently it is. Stable enough for coral timescales.
That's the first big one we've seen.
Ooh. I think I can answer this question in chat. How do you know you aren't sampling DNA from multiple locations since the Niskin bottles are open with flowing water through them all the time? You would think you need discrete samples.
Roger.
Well, with the Nitskin bottles, they're currently open, but as soon as we close them, it becomes a closed system and the water inside stays inside. And then we can go to other sites and close different Nitskin bottles and the water is going to be different. So when the Nitskin bottles are currently open, the water flows through. When we close them, the water is sealed inside, and then when it reaches the surface, of which Meredith, who was on, just was in place of Steve, goes in there. She gets these cool-looking bags that you fill up, and then you filter them through a water filtration system in the wet lab. Steve,
Very
did I get
nice.
that right?
Yep. This is a possibly bathypathes or telopathes. It's one of these branching bathypathes looking species, but I think we had called this either telopathes or something very similar. But I know I've sampled this one before because I've seen this and I was astounded the first time and I'm not so surprised now, but yeah, it's very different, unique morphology.
And is that a, what kind of, is that a squat lobster?
Yeah, it's a Euplectilliid squat lobster. 3 of them. Yeah, so this is possibly a telopathes, but it also could possibly be a bathypathes. It's got 3 bench points, which is kind of unique.
Do you want a close-up?
Yeah, sure. Actually, if you could give a close-up of the branch point here, let's see what's up.
Go ahead, Dan.
We're not going to do any sampling, but it would be nice to get a peek. Looks like it has 1 solid branch that comes off, and then all these pinnules.
Let's see, I'll have to go wide. I lost the plot where the branching was. Can you go wide?
Sure.
This is one of the biggest ones I've ever seen, though.
Okay. Now I got it. You can zoom back in again.
Yeah,
Have we seen black coral larger than this yet?
Larger than this? No, we have not. There are not many that are larger than this.
So large.
Yeah, maybe the occasional Liopathes might be larger than this, but this is a pretty good size one. But it has this characteristic Europtychus, very spiky Europtychus, which seems to be unique to this particular species of black coral.
There's a fish there. I was just going to say there's a...
There it is.
Sorry, what are you seeing back there?
Fish right here. It's flowing past us. All good.
Yeah, I'm gonna come up for some clear water here.
And which coral was that again? Was that a heteropathes?
Possibly bathypathes or telopathes.
Let me know when you're okay for
Target
gauge,
it for
gauge checks, Dan.
folks on shore to make
Let
that
me know
call.
when you're okay for gauge checks.
Can you full screen that?
Yeah.
It's
Yeah.
Some of the candelabra corals.
They saw, wow, 1.5 meters. There's some I-4 clade bamboo corals here too, the candelabra type bamboos.
Another Paragorgia.
Yep, Paragorgia.
Bathypathes.
Yeah, so we've got bathypathes here. One of them looks very similar to bathypathes pseudo-alternata, like this one down here. This one was recently described, recently re-described or replaced just a few months ago. A paper came out. But then there's also this heteropathes or parantipathes. I'm not sure what we were calling that, but we sampled it extensively last year. Has characteristics of both heteropathes and bathypathes, or heteropathes and parantipathes, but I think we'll stick with heteropathes on that one.
And is that a I4 clay bamboo coral you were seeing up there?
That's right, in the big one on the left. Yep. And a red Parysocrinus crinoid.
all right. Come back around on the point here.
So that question in chat, as the follow-up question to the Netskin bottle, aren't trace DNA being left behind inside the surface of the Niskin?
Okay, to take the bubble for a sec.
So you can't say it came from a given site
Sure.
with much certainty. I'm not exactly sure how it's done. I'm not a full-on expert on the exact how the process works. Meredith is the person to go to that.
Gauges look good.
But the way that I'm sure the Niskins work
good for 20.
is they capture enough water from there, and then when it filters through,
I think so.
it's and when they, I'm not sure. the long end, the short end of that question is I'm not entirely sure. There's ways that they do it that they can account for that and filter out the common DNA that they find inside the Niskin bottles, but I'm not exactly sure the exact process.
There is a way of doing that on the back end, but if two samples look curiously similar, there are ways to clean the data and get it. But for the most part, you know, a lot of the microbial material, you know, that's probably what you would imagine would be sticking to the sides of the jars or something like that. All of that is actually filtered out completely. It's not filtered at all by our eDNA filters. So it's really not that much of a concern. But you know, if two samples do look curiously similar, we have ways to fix that data set.
We have ways of fixing it.
We also take a lot of water samples, so if something looks erroneous, like if it's showing us species that are very dissimilar from what we observed, we can omit pieces of data. Bottles malfunction sometimes, and that's why we have cameras that are watching them all the time to make sure that they're not leaky or anything like that.
And when you pour them, you have to be very, very careful you do not spill any or get any on like the outside of the bag, because it's very, very, very easy to cross-contaminate. And when handling the samples, little behind-the-scenes fun fact, we use this solution called DNAOA, which is something it's stronger than bleach, but not as harsh as bleach, that you have to scrub all over your gloves and your forceps that you use.
Synaphabranchidiol?
Branchidiol.
And it's basically hand sanitizer that gets rid of 100% of the germs.
Oh, wow, good eye. Branchidiol.
Good for another move. Let's do this next move, 045. Yeah.
Hello, fish.
You said it was a branchidiol?
That's a synaphabranchidiol, yeah. Synaphobranchid. Yeah. Cutthroat eel.
Okay.
Synaphabranchidiol. Nice.
Why didn't you say that on SBL?
I'll say it again. I totally stole it from chat. But see, Steve here knows a lot about coral, and I asked him, would he consider himself a prim know-it-all?
Yes.
Because he knows a lot. Thank you, person in chat. I totally stole your joke. But thank you.
Have you heard that one before?
I haven't heard that one before.
Wow.
Hey.
So I imagine we want to pick up a rock eventually, if we can.
I haven't seen much broken stuff since we got back.
It seems to be getting steeper, so we might not have a chance until we get up on top of this little platform.
Looks like a bamboo with a Venus flytrap anemone on it.
Yeah, mostly it's probably in the 10 to 20 species just on this little, since we started getting into the columnar territory. It's kind of the depth where you start seeing a lot of the high density or high diversity bathyal community. Bathyal usually between between about 200 meters all the way down to about 3,000 meters. But sometimes you can have different diversity maxima within the bathyal zone. This one I would call it pretty representative of the deep Central Pacific bathyal diversity maximum community. We've done some diversity analyses a little bit further south in the area around Howland and Baker Island, and there does seem to be a deeper bathyal diversity maximum and then a shallower one. Whereas in the Atlantic, usually you have just one mid bathyal diversity maximum around 1,200 meters. Here you have one between 1,800 and 2,200 usually, and then you might have another one around 12 to 1,400 meters.
Is that a sea urchin over there?
Uh,
Sorry.
there's bathypathes, or even next to the lasers.
Yeah.
Yeah.
Or like it just like went right below it, yeah.
That's a crinoid.
Oh my gosh. I need new glasses, sorry.
Yeah, 045, kind of keep us on the point here. It goes off.
What's the current like around here?
Left to right, to the east. Seems like more of the critters are on the east side here.
Huh, towards the east.
I think so.
That's unusual.
It's the opposite of the...
Yeah, the models suggested out of the, from the east would be the current direction, but it's not. It's happened before where, you know, deeper below kind of these promontories, these platforms, it's one direction, then it swaps around and comes the other way. There's Victorgorgia right there.
The model's a lie, the cake is a lie.
Well, their models are notorious for this saying that most or all models are wrong, but some are useful, and that's true.
It seems like there's more animals. I mean,
And
right
most
here
models will
locally around
perform
the
exactly as you expect when you put, you know, various quality data sets into them. So if you put poor quality data sets into a model, you get poor quality out. So generally, you know, if you have the ability to do like ensemble modeling or something like that, where you have lots of different, you know, model algorithms running, and you can combine and take the statistically the most appropriate one for your needs, that usually provides you the best amount of certainty. But we've been playing with the model set. Rob's been running all models for these sites we've been visiting, and we seem to have gotten it down to a few variables that are kind of causing some confusion in the model, and it's actually been performing pretty well. These are models largely based on just oceanographic conditions and water flow, pressure gradients in the ocean, so it's impressive without any, you know, measured data, really. The only way to really be sure would be to have something like an ADCP or something mounted on the hull, which would really be a really fantastic piece of equipment that will tell us the water column current speeds at various different bins through the water column.
Do we have plans to attach those sort of things to Hercules or Argus or something?
I don't know.
There is a rumor we're going to stick one on the boat for next year.
Ooh. So tune in next year, Nautilus Live viewers, or stay tuned until next year.
Just a ROV rumor, though. I don't know. I don't know what the real deal is. We would like one for non-scientific reasons, so on our wish list.
For recreational use.
It helps us to determine what the current's doing. When we're launching recovery in the ROV, we can plan accordingly.
Atlanta heading 040.
Right.
So Steve, I was wondering about colors of corals in the deep sea. You know how like animals in the shallower ocean, like sharks for example, have adapted to have these colors that help them sort of predate, and they blend in from below and from above.
Yep.
Are the colors in the deep sea an adaptation of some sort, and are they beneficial, or it's just so shocking to see so many bright animals in such a dark place?
Well, I mean, the first answer most people go to is that, you know, warmer colors will tend to attenuate faster in the visible spectrum. You know, so if you have, you know, warmer colors like reds, yellows, oranges, those will, you know, any light that's available will, you know, attenuate those wavelengths pretty rapidly, such that, you know, they appear gray or black or, you know, shadows. But, you know, I would wager that most animals probably don't use, you know, a very broad visible spectrum. They're probably very specialized at this depth. So, you know, you might see things that are probably more
next move by
seeing in the bluer range if they're using visible light
a bit more to the north there.
or other wavelengths entirely.
Yeah.
So, decent rock pile.
Yeah. Don't know. Yeah. I assume the Atlanta doesn't want to be right there.
Yeah.
So you want to rock from here, do you?
Yeah.
Grab one up there, Gibson.
Roger.
Yeah, if you guys can maneuver it. Absolutely.
I got it. Help me with the sonar there, Lynette. I got a cliff just to my left and right ahead of me.
Yeah, she's moving the boat over that way.
Roger.
If you'd like to click over to channel 30, you can see our very own Gibson take a crack at getting a rock sample. Crack, rock. Yeah.
Nice. All right, craft is live.
Let's see.
There's a little poke right here.
See anything angular?
I'm thinking just to my right.
Yeah, like, I don't, that looks connected, but maybe not.
Yeah, it's connected, but just below it. Wow.
Oh, yeah. Oh, okay.
And the hull button's not working.
Yeah, the one you're touching right now seems okay. Seems good.
Don't halt then.
Well.
Pick up a rock. That's one way to do it.
Roger.
Yeah, that one right there is blocky.
Let's do that then.
Yeah.
Starboard box.
Yep.
Yeah,
looks great. Yep, looks great.
All right, so we're going to go in the starboard box.
Roger that.
Can put it right in bin A.
Got some dust on this bad boy.
Ready?
Yep, ready.
Nice.
Box A.
Awesome, thank you.
Well done.
Seems like the halt button's working just fine.
Halt button's working great.
Oh, I thought you said it wasn't working.
I did, I just had a moment of missing the halt button. All right.
It's kind of like I went to the doctor and I said, doctor, when I lift my arm up like this, it hurts. And the doctor said, well, don't lift your arm up like that.
Number of times my dad's told me a joke like that. All right, let's stow this guy.
Great. We have some questions about the whale skull
Is that all right there?
that we found the other day.
Yeah, that's fine.
Yeah, works great.
There's a question about the whale skull you sampled. Was it fossilized, or did the ocean interfere with the fossilization process?
You know, we really don't know all that much beyond what's on the outside surface. So the fossil was completely crusted over with iron manganese, so it won't be until we really can get a chance to look at the interior by potentially removing some of that manganese or figuring out what's underneath, will we really know what effect all that time at depth has had on the piece.
Raj, let's hold here for a minute. Let me pop up over the ridge.
Crabs.
Not seeing anything on that sonar for a while, so
And then
don't see anything on Atlanta sonar, like except for to the south. So I don't know where we're supposed to be going.
And yes, Jason. Yes, it is. You're
That's
right.
really precarious.
Wow, what a life-filled rock.
The animals love that one.
Yeah.
I think we're just at
It's
the
protruding.
we're just at the top of the feature here.
Oh wow! Look at that. It looks oddly. You guys ever see the new Stars movie? And you know that the crashed Star Destroyer, where it's all covered with moss.
Let's move 20 meters to
That's
the
what it
north.
reminds you of?
Or
Yeah.
poke around the top here. Yeah, this will probably be the last move
I was
north.
going to say the Lion King Pride Rock. That was a nice lillipathes in the background, possibly. A black coral. Lillipathes or chrysopathes. We'll take a look when we get closer. Talking about this one here.
Is this like an example of like
Yeah,
recruiting?
Raj, we have to move the
Yeah,
ship.
I think there's no. I don't know to what extent this has to do with reproduction. I think it might just be a popular spot. I mean, they could be recruiting nearby, but usually when you find promontories like this, animals will gravitate naturally towards the areas of better flow. It's something we see typically. That the best spots are always colonized by the greatest density of animals.
A large
So-
crab right there.
These.
That's a Brisingen sea star.
Is it really?
Yeah, I did. I got a quick peek at it
Oh,
Right.
wow.
before we were talking about this down here.
So Steve, someone in chat wants to know these coral. How long does it take for them to grow about 10 centimeters, or is it just too hard to tell?
So they're probably growing at. I'll take a stab at it because every group is different, but at this depth they're probably growing on the order of maybe millimeter, couple millimeters at the very top end, maybe every year or 2 years. So not fast. That would be a very fast growth rate. I think it's probably more like millimeters per decade, something like that.
So by that
Or irrdigorgia.
Yeah, and another paragorgia behind it.
Nice spot.
With some branching bamboos.
Wait till you see.
So
Wait till you
person
see
in
the
chat,
backside.
Yeah, this would go against the model, I guess, because the flow seems to be in the opposite direction. But they were like weighted, the flow, right? So.
Yeah, I wouldn't be surprised. So one of the things that we found at one of our other sites was that even though we had kind of a eastward or a current from the east flowing, as the predominant bottom current, if it wraps around and it shoots off, swirling eddies around the back end of these promontories, that can sometimes cause reverse flows. So it's not unobserved. We've seen something similar. I think the topography here is just absolutely got to be a factor, though. This stuff is really stable, a lot of angles, a lot of potentially accelerated flows. So I think all around it's a great spot.
Very cool.
In your bubble, you can just see how much life is down there.
Yeah,
So
it's
Steve,
tempting to go down there and poke
from
around.
this watch, are we
Yeah.
hoping to collect anything specific?
You know, same objectives as always. So we're out for, you know, interesting or curious or unusual biology. But in addition, you know, rocks and water samples for eDNA, as well as, you know, if we see any sea cucumbers or echinoderms, you know, that might be predating, for example. We are hoping to sample those too.
Was this the one you wanted to?
Yeah, the orange one there. Yep.
And nodule fields if we see them.
Nodules too. Yep.
Should have got a rock from here.
Yeah.
Not too late.
Looks like a rock beach on the left.
Totally.
I have to tilt down here.
Trying to get it in the.
There you
see.
are.
Yeah, Kylie said the tilt wasn't working too well.
Yeah, I agree. It's pretty sticky.
Do you want to zoom on this guy, Steve?
Yeah, if possible.
Yeah, go ahead.
Sure thing. Is a half zoom?
Yeah, you should be able to full zoom there.
Sure.
Perched on something.
That's full.
Sorry, I slipped. Let me try that again.
It might be my tether. That looks a lot like a pine tree to me, a red pine tree.
Kind of does.
Okay, try again.
Sure.
You know, I'd like to take a sample of this, if we can.
Roger.
Some kind of black coral?
Yeah, I'm between a couple of different genera. I'm leaning towards Chrysogorgia, but a small piece would help us better identify this. Good, thanks for the focus.
Yeah.
So why are we having some trouble identifying it? Are there different features that stick out that you would find odd?
Yeah, I'm more specialized in sea fans, and black corals are a little bit different, and we have some specialists ashore who can help us identify this in better detail, some folks who are specifically focusing on black corals. So Dan, if we can get at least one branch point, that would be ideal. We don't need a big piece, you know, under 10 centimeters, I think, if we can find the right bit.
Got a T at the top there.
Yeah, that might work. Yep.
Right in there. Is that enough? Those three little branches?
Yeah, that would be fine. Yeah, that's
more if you want.
that's under 10. Yep.
Right there.
Great.
Nice.
We can do bio box, but there's a floaty sea cucumber.
Yeah, can we go to the starboard box for this one? This one actually could be lillipathes too. Okay, good capture, and we're going to go to the starboard box.
Okay,
We can put it.
for a second.
Sure.
We can put it in A, the same box we put the last one, the rock.
Roger.
My gut is telling me lillipathes, but I know there's something about lillipathes not being very common out here.
Tilt up on bubble there.
Roger.
See if I'm going to clear the rock. I'm going to have to move.